Hello students, welcome to EPG Pathshala. I am Dr. Kapendra from Kirodimal College, University of Delhi. Today we are going to discuss about a module Morphology, Life Cycle, Mode of Infections of Trypanosoma under paper Biology of Parasitism. After studying this module, you shall be able to know the disease human African trypanosomiasis, learn different forms of human African trypanosomiasis, know the distribution and major epidemics of the disease, understand the life cycle of Trypanosoma Gambiense, learn the mode of infection, symptoms, diagnosis and prevention of the disease. Human African trypanosomiasis, also known as sleeping sickness, is a vector-borne parasitic disease caused by a protozoa which belongs to the genus Trypanosoma. The parasite is classified into three subspecies: Trypanosoma gambiense, Trypanosoma rhodesiensis, and Trypanosoma brucei. The latter subspecies is not pathogenic to humans. These subspecies cannot be differentiated morphologically. Trypanosoma gambiensis is distributed in Western and Central Africa, which causes chronic disease, whereas Trypanosoma rhodesiensis found in Eastern and Southern Africa and responsible for acute severe disease. The epidemiology of these parasite species also differs and depends on the distribution of their vector Glossina palpalis and Glossina morsitans respectively. The Glossina palpalis, found in the areas of vegetation near rivers or cultivated fields. The Glossina morsitans mainly depends on wild animals in savanna which are far distant from human settlements. The disease is transmitted to humans by tsetse fly, which got their infection from humans or from wild animals harboring the parasites. Some species of trypanosomes are non-pathogenic and does not cause any harm to the human, while other species such as Trypanosoma gambiense are pathogenic and cause several diseases in human and animals. In this module, we will discuss about morphology, life cycle and pathogenesis of Trypanosoma gambiense. We will also take a glance of diagnosis and treatment of the disease. Forms of human African trypanosomiasis. Human African trypanosomiasis takes different forms depending on the parasite involved. Trypanosoma gambiense is found in West and Central Africa that currently account for around 95% of sleeping sickness cases and causes a chronic infection. In this form, the individual infected with the parasite can live without any symptoms of disease for several months or years. When a symptom first arises, the patient is already in an advanced state of disease where central nervous system is affected. Trypanosoma rhodesiensis is present in eastern and southern Africa. This form represents 5% cases and causes acute infection. The symptoms of the disease appeared within few months or week after infection. The disease occurred rapidly and invade the central nervous system. Trypanosoma cruzi occurs mainly in about 21 Latin American countries. It is called American Trypanosomiasis or Chagas disease, the causal organism is a different species from those causing the African form of the disease. Trypanosoma rangeli is a non-pathogenic trypanosomes found in the blood of men in Venezuela and also in Colombia. Distribution and major epidemics of the disease. The disease sleeping sickness is endemic and highly prevalent in some regions of sub-Saharan Africa, covering near 36 countries, having more than 60 million people. Many of the affected populations live in remote areas having limited access to adequate health services, which obstructs the surveillance and hence diagnosis and treatment of the cases. Despite its sporadic occurrence among travelers, TB rhodiensis has been found more common in European and American tourists than TB gambiensis because rhodiensis is present in areas not visited by emigrants. According to recent study, total African population at risk of sleeping sickness is 69.3 million, with one-third population being at very high to moderate risk and rest of the population at a low to very low risk. About 48,000 people died of sleeping sickness in the year 2008. There were four major epidemics occurred in recent history in Africa. First epidemics occurred mostly in Uganda and Congo Basin between the year 1896 and 1906. Two epidemics in 1920 in most of the African countries. Third epidemic occurred in 1970 in several African countries. Fourth epidemic occurred in 2008 in the Uganda. WHO established public-private partnerships with Aventis Pharma and Bayer Healthcare, which support epidemic countries in control efforts of disease and supply drugs without any cost for the treatment of the patients. The efforts of WHO, bilateral cooperation, national control programs and non-governmental organizations during the 1990s and in the early 21st century has stopped and reversed the upward trend of new cases of the disease. Trypanosoma gambiense. Trypanosoma gambiense as a human parasite was first discovered by Ford in 1901. Sir David Bruce in 1895 first found that the tsetse fly is responsible for transmitting the disease sleeping sickness. Trypanosoma gambiense is confined mainly to Central and West Africa, particularly in Nigeria and Congo. Areas near the rivers and lakes have the greatest incidence of infection since the insect vector tsetse fly lives there in low and thick vegetation. Trypanosoma gambiense are microscopic, elongate, flattened and have fusiform body pointed at both ends and covered by a membranous pellicle which maintain the form of body. It measures about 10 micrometer to 40 micrometer in length. A single flagellum arises from a basal body situated near the posterior end and curves in a spiral form round the body, forming undulating membrane thrown into 3 or 4 folds depending upon the length of the parasite. The undulating membrane is believed to be an adaptation for locomotion in the blood. The flagellum is free at the anterior end. The nucleus is large and oval situated in the center of the body, and the cytoplasm contain numerous greenish refractile granule called volutin granules. These granules store food particles, mainly glycogen and phosphate. At the base of the flagellum is located the basal granule, close to which is another granule, the parabasal body. Life cycle. Trypanosoma gambiense is a digenetic parasite which require two hosts for completing the life cycle. The primary host is human and the intermediate host is blood-sucking insect tsetse of the genus Glossina. The mammals like pig, antelopes, buffalo often acts as reservoir host harboring the parasite. When tsetse sucks the blood from infected individual or wild mammals, it carries trypanosomes to its midgut where they divide asexually by longitudinal binary fission. Here, the parasite changes their morphology and give rise to metacyclic forms which are short and stumpy. At this stage, the fly is said to be infective. When the infected tsetse bites a healthy human host, it releases these metacyclic trypanosomes into the bloodstream of the host and repeats the life cycle. Sexual reproduction is unknown in Trypanosoma gambiense. It is essentially a parasite of connective tissue in human where it multiplies readily. It consumes large amount of glucose and invade the regional lymph nodes through the lymphatic system and also invades blood system causing parasitemia. It mainly localizes into brain. It is to be noted that African sleeping sickness is a disease which affects the central nervous system. Life cycle in human. The infection by parasite is initiated when tsetse harboring the infective metacyclic forms bite the healthy individual. When the fly bites, it releases trypanosomes into bloodstream which develop into long slender form and multiply asexually by binary longitudinal fission at the site of inoculation. These become stumpy via intermediate forms. Consequently, the parasite invades the bloodstream and causes parasitemia. The trypomastigote forms, mainly the short stumpy forms, are taken up by the tsetse along with its blood meal and undergo into a series of complex biological development inside the insect host before becoming infective to the man. All stages of parasites in human are extracellular as they are present in the blood cell. In human blood, the metacyclic forms which are devoid of free flagellum become transformed into long slender forms equipped with long flagella. These stages can freely swim by beating of their flagellum along with the vibratile movement of undulating membrane. They multiply asexually by longitudinal binary fission. The multiplying trypanosomes obtain energy by anaerobic process of glycolysis. When absorption of glucose ceases due to antibodies which are produced in the blood hamper the glycolysis, as a result, trypanosomes stop dividing and shrink to the short stumpy forms, which are lacking free flagellum. These stumpy forms do not feed and ultimately die if they are not sucked up by tsetse along with the blood meal from infected human. Life cycle in tsetse. When tsetse feeds on the blood of an infected person, it also sucks short stumpy forms of the parasite along with the blood. It is the stumpy forms which continue development in the vector. Further development of stumpy forms proceeds in the insect midgut within peritrophic membrane. In the midgut, parasite transforms into long slender form and multiply asexually by longitudinal binary fission. The kinetoplast moves further from the posterior end of the body. The energy yielding process is related to mitochondrial oxidation of pyruvic acid. After some time, the long slender forms migrate into salivary gland via the esophagus and mouthparts of the insects. Here they are metamorphosed into the critidial forms which with shortened body, reduced free flagellum and the kinetoplast in front of the nucleus. The mitochondria develop an extensive network of cristae and parasite respires more economically as blood glucose gradually declines. The critidial forms multiply in the lumen of salivary glands and transform into slender metacyclic forms. When the tsetse fly bite a healthy person, it transfers the metacyclic stage into his blood where they initiate another infection. Mode of infection: inoculative method by the bite of the infective tsetse fly Glossina. They bite the daylight, usually in the early morning and evening. The metacyclic stage is introduced by the tsetse fly with the saliva into the subcutaneous pool of blood on which it feeds. Some of the parasites may enter the bloodstream directly and majority of them entangled in the tissue space. The initial growth of trypomastigotes occurs in the tissue space which form a favorable nidus, or possibly here the organism can escape the action of antibodies which might be developed. It is to be noted that while the trypomastigotes are multiplying in the subcutaneous tissue, the organisms are either absent or present in small numbers only in the peripheral blood. It has been suggested that although unlikely that the connective tissue damage caused by the trypomastigotes may be due to an exaggregated immune response rather than to any direct effect of these relatively non-toxic organisms. The presence of trypomastigotes in the subcutaneous connective tissue excites host's immune response in two ways. A. By producing large amount of non-specific immunoglobulins which are however not capable of sensitizing the antigen. Antibodies are produced in response to the secretion of an exoantigen of the trypomastigotes. By heavily infiltrating the site of infection with macrophages, the cells competent to deal with the invaders. The neutrophils take peculiarly little interest in the defense and are therefore not much in evidence. Thus, it will be seen that there is no lack of mobilization of the host's defensive mechanism, but it is the cellular defense which plays the dominant role. The macrophages could be seen to remove the living trypomastigotes in the tissue space. The release of kinins may help to attract macrophages. It also increases the capillary permeability of tissues and may explain the edematous swollen subcutaneous tissue at the site of infection. Furthermore, trypanosomes are surrounded by a coat that is composed of variant surface glycoproteins, that is VSG. These proteins act to protect the parasite from any lytic factor that are present in human plasma. The host's immune system recognizes the glycoproteins present on the coat of the parasite, leading to the production of different antibodies like immunoglobulin M and immunoglobulin G. These antibodies will then act to destroy the parasites that circulate around the blood. However, from the several parasites present in the plasma, a small number of them will experience changes in their surface coat, resulting in the formation of new VSGs. Thus, the antibodies produced by the immune system will no longer recognize the parasite, leading to proliferation until new antibodies are created to combat the novel VSGs. Eventually, the immune system will no longer be able to fight off the parasite due to the constant changes in VSGs, and infection will arise. Clinical features: bite of tsetse fly causes local irritation which subsides after few days. A trypanosomal chancre may develop at the site of inoculation of trypomastigotes introduced by the bite of the infected tsetse fly. It is a hard, painful nodule, and fluid withdrawn from it contain actively dividing trypomastigotes. It subsides in a week or two without suppurating. The system can be appeared after several months or a year in Gambian forms, and symptoms may appear after 2 weeks in case of Rhodesian forms. It is characterized by the infection of bloodstream, involvement and enlargement of lymph node, and eventually invasion of the central nervous system. The early symptoms are fever, loss of nocturnal sleep, severe headache, and feeling of oppression. A fleeting circulate erythematous rash may appear on the chest and shoulder. Lymph node enlargement, particularly of the posterior triangle of the neck, is a feature of Gambian disease, whereas invasion of central nervous system is very rapid in case of Rhodesian forms. As the central nervous system is involved, the symptoms of meningoencephalitis develop, resulting in classical sleeping sickness. In due course, the patient fall asleep first at regular interval and then lies prostrate in coma. Finally, the patient becomes thin and exhausted, accompanied by sign of malnutrition. Disruption of the sleep cycle is an important symptom of this stage that gave the disease the name sleeping sickness. The person infected from disease experience unsystematic and uneven 24-hour rhythm of the sleep-wake cycle. The patient sleeps in daytime and at night time shows period of wakefulness. Other neurological symptoms of the disease include tremor, confusion, paralysis, general muscle weakness, hemiparesis, and paralysis of the limb. Parkinson-like movements may also arise due to non-specific movement and speech disorders. The person infected from sleeping sickness may also exhibit psychiatric signs that aggressive behavior, irritability, psychotic reaction, or apathy, which can sometimes dominate the clinical diagnosis. If the disease is not treated, it can invariably become fatal, with progressive mental deterioration that lead to coma, failure of systematic organ, and finally death. In case of TB rhodiensis, an untreated infection will lead to death within few months. However, infection with TB gambiensis will lead to death of the patient after several years when left untreated. Tryptophan is a chemical compound which stimulates sleep in humans. It is the chemical that is produced by trypanosomal parasite in sleeping sickness. The major role of transmitting the disease is bite of an infected tsetse fly, but there are several other ways through which people are infected with sleeping sickness. That are, the infection can be spread from pregnant mother to her child because the trypanosomes are able to cross the placenta and cause the disease to the fetus. The mechanical transmission is also possible through other blood-sucking insects. However, assessment of epidemiological impact of transmission is very difficult. Accidental infection may also be possible in the laboratories due to pricks from contaminated needles. Disease management can be done in three major steps. The first step is the screening for potential infection which can be done by serological test and confirms major symptoms such as swollen cervical glands. The second stage is to diagnosing the presence of the parasite. The last step is the staging, which is done to find out the stages of disease progression. This involves collection and examination of cerebrospinal fluid from lumbar region, which helps in determining the course of treatment. The Gambian or African trypanosomiasis or sleeping sickness can be treated during early stage, but once the parasite enter into the cerebrospinal fluid of central nervous system, it is very difficult to control the disease. There are number of drugs that are all used to control the disease. Drugs such as suramine sodium, Bayer's 205, atoxyl and trypanosamide have been useful in early stages of infection. Other drugs such as parsenophenyl butyric acid, germanin and pentamidine are also effective in treatment of early cases. Orsenine is quite effective in cases where the central nervous system is involved. Millarsen oxide is rapid in action and is less toxic. Prevention depends upon the eradication of vectors, that is the tsetse fly. It can be eradicated by destruction of the habitat of the vector. It is supplemented by the use of insecticides such as spraying of DDT over bushy areas in the vicinity of villages. Game destruction program to eliminate the blood meal of the fly and isolation of the human population from areas known to harbor infective animals can be done to prevent the spreading of the disease. This is much more important in case of Gambian disease. Pentamidine is effective against Gambian disease. A single intramuscular injection of 4 mg per kg will provide a preventive effect for at least 6 months. So students, let us summarize what have we learned in this module. Human African trypanosomiasis is a vector-borne parasitic disease caused by the protozoa which belongs to the genus Trypanosoma. The disease sleeping sickness is endemic and highly prevalent in some region of sub-Saharan Africa, covering nearly 36 countries having more than 60 million people. There were 4 major epidemics occurred in recent history in Africa. Trypanosoma gambiense as a human parasite was first discovered by Ford in 1901 and is confined mainly to Central and West Africa, particularly in Nigeria and Congo. The parasite initially lives in the bloodstream and lymph glands of the patient and later invades the cerebrospinal fluid of the CNS system. The parasite is transmitted by vector Glossina palpalis. Trypanosoma gambienses are microscopic and covered by a membranous pellicle. It also bears a single flagellum which curves in a spiral form round the body, forming undulating membrane adapted for the locomotion in the blood. Trypanosoma gambiense is a digenetic parasite which required 2 hosts, human and tsetse fly, for completing the life cycle. When tsetse fly sucks the blood from infected individual, the parasite divide asexually in the midgut of the insects by longitudinal binary fission and forms metacyclic trypanosomes which are infective and ready to infect new hosts. When the fly bite, they invades the regional lymph nodes through the lymphatic system and also invades the blood system, finally localizes in the brain and affect central nervous system. Lymph node enlargement, particularly of the posterior triangle of the neck, is a feature of Gambian disease, whereas invasion of central nervous system is very rapid in case of rhodesian forms. The symptoms of meningoencephalitis develop, resulting in classical sleeping sickness in which the patient falls asleep in initial stage at regular interval and then lies prostrate in coma. Disruption of the sleep-wake cycle is an important symptom of this stage that gave the disease the name sleeping sickness. There are number of drugs that are used to control the disease. Drugs such as suramine sodium, Bayer 205, atoxyl and trypanosamide have been useful in early stages of infection. Other drugs such as parsenophenyl butyric acid, germanin and pentamidine are also effective in treatment of early cases. Orsenine is quite effective in case where the central nervous system is involved. Millarsen oxide is rapid in action and is less toxic. Thank you.